Hello, this is Team Fischer Esterification, and our research focuses on class imbalance and false inferences in breast cancer spatial transcriptomic classification. Our research question focuses on to what extent does class imbalance in a slide sequence V2 breast cancer spatial transcriptomic dataset contribute to false inferences when classifying malignant cells versus non-malignant tumor microenvironment cells. Our hypothesis stated that severe class imbalance in breast cancer spatial transcriptomics compels machine learning models to yield elevated rates of false inferences, thereby degrading malignant classification accuracy within highly heterogeneous tumor environments. Some background is that breast cancer is a leading diagnosis in the realm of cancer screening in women, and early detection can become the primary driver of survival, as women who are screened regularly have a 60% lower risk of dying. Machine learning tools can assist in creating new and improved diagnostic tools to help alleviate the pressure on pre-existing mechanisms of detection and further aid in reducing the mortality rate of breast cancer. This research aims to understand how disproportions in spatial transcriptomic databases can ultimately lead to false inferences in the characterization of malignant breast cancer cell states compared to non-malignant tumor cells within the context of the tumor microenvironment utilizing machine learning classifiers.
For our methods, we used a breast cancer slide sequencing V2 dataset from the CZ Cell Exchange portal. The dataset included 5,833 annotated observations and 12,630 gene expression features from one donor sample. Using Python in Google Colab, we created a binary outcome. Observations labeled as malignant were assigned to the malignant class, while specific cells labeled as non-malignant were placed in non-malignant tumor microenvironment, or TME class. For pre-processing, we normalized the raw count matrix to 10,000 total counts per observation, applied a log transformation, and selected the 2,000 most highly variable genes. These processed features were used for PCA visualization, logistic regression, and feature importance analysis. For model comparison, we used a majority class baseline that predicted every test observation as malignant. We then trained a logistic regression classifier using a stratified 75-25 train test split. The results showed that 94.77% of observations were malignant, while 5.23% were non malignant TME. In the PCA plot, malignant observations formed the main cluster, while non malignant observations were fewer and appeared both within the main cluster and as scattered separated points. The baseline model had an accuracy of 94.79%, but it did not identify any non malignant TME observations. Logistic regression had an overall accuracy of 91.30%, a balanced accuracy of 64.32%, and correctly identified 26 out of 76 non malignant observations. The feature importance plot showed the genes with the strongest positive and negative coefficients for malignant and non malignant classification.
To reiterate, this study explored whether class imbalance in a breast cancer spatial transcriptomic dataset leads to false inferences when classifying malignant spatial transcriptomic observations versus non-malignant TME. These findings seem consistent with general and broader concerns where models may appear accurate due to the majority class dominating it, while the minority class errors are hidden by the overall accuracy score. An important implication of this study is that transcriptomic classifiers should not be judged only by their overall accuracy in imbalanced datasets. If minority populations are poorly detected, a model can appear reliable while it fails to identify vulnerable and underrepresented cell populations. This project came across several limitations. One, the analysis used one cell sequence V2 breast cancer dataset. 2. The dataset only contained one donor sample. 3. The binary grouping of all non-malignant TME cell types into one class simplified a biologically diverse group. And 4. The model depended on the already existing cell type annotations, with errors influencing both training and the evaluation. Future research direction should focus on various spatial transcriptomic or single-cell RNA sequence datasets with more patient data or a better TME representation. In conclusion, model evaluation metrics and interpretability methods are important in identifying possible false inferences in machine learning models used for cancer transcriptomic classification.